Hello everyone. Today we're going to use Rebel, the classroom bioprinter, to print simple algae-based arrays to study photosynthesis. In order to do so, we first need a biomolecule that can carry out photosynthesis, aka convert CO2 to oxygen and other products. So here we have green algae. We also need a biomaterial that's able to encapsulate algae into the array format, and we're going to use Agarose, which encapsulates algae and allows us to print algae in array format. And once we have printed algae, we also need a means to study photosynthesis or visualize the reaction proceeding. For that, we have the bicarbonate indicator. The bicarbonate indicator is a pH-based indicator. CO2 in solution form is a weak acid, and as the photosynthesis reaction proceeds, more of the CO2 is being captured by the algae to lead to a change in pH. So over here, we have the solution when the CO2 has not been captured, so you can see a bright yellow color. And as we proceed to the left, more CO2 is being captured, and that leads to a change in pH and a purple color. And therefore, we can use the bicarbonate indicator to study algae-based photosynthesis and conversion of CO2 into oxygen. Okay, now we're going to move to the second phase of our demo. We're going to use the bioprinter to create algae-based arrays. For that, we're going to mix algae with agarose to create this first syringe. We're doing this so that agarose is able to encapsulate algae and prevent it from escaping the array once it's printed. Next, we're going to also create the second syringe, the bicarbonate indicator, in order to visualize the photosynthesis. As I mentioned before, when we add the bicarbonate indicator and as the CO2 is sequestered, you will see a change in color from yellow to purple. Okay, now we load the algae syringe for the first print. The first print is done, so we're going to proceed and unload the algae syringe and then load the bicarbonate syringe. As you can see here, the bicarbonate indicator is being layered directly on top of the algae droplets. Our prints are completed. Now we're going to wait for 4 hours to see a change in color. As you can see, it's now about 4 hours since we printed the array, and the color has changed from yellow to purple.